Harnesstom is a strong coalition of scientists, breeders, and farmers. We are working in research institutions, in seed companies, and NGOs of 7 countries to conduct a project entitled Harnessing the Value of Tomato Genetic Resources for Now and the Future. In Harnesstom, we are convinced that the amount of genetic resources available for tomato and the knowledge that has been developed over the years in the EU and elsewhere can be put to work to solve a number of issues that are threatening tomato cultivation worldwide, and which include emerging diseases, climate change conditions, etc. We are also trying to satisfy the changing consumer preferences towards more flavorsome, more nutritious and healthier tomatoes. Our approach is knowledge-based, so the first thing has been to centralizing the scientific information available, and then by developing ring-fenced targeted pre-breeding programs, where specific breeding companies and academic groups are working together towards specific objectives of interest. This approach, we believe, takes advantage of each of the companies' specific interests and knowledge, and facilitates the exchange of information. On the other hand, we're also conducting participative breeding directly with farmers to increase resilience and introduce innovation in traditional tomato while paying attention to consumers and the rest of the stakeholders. The project is progressing pretty well, and details on the progress will be given by the Work Package leaders, but some highlights are the construction of the largest database on tomato genetic resources information, which is well on the track. So it is the development of pre-breeding tomato materials to increase tolerance to heat, drought, and salinity and combinations. We have, for instance, a clear map and a strategy to control new strains of Cladosporium fluvium that have overcome the current resistances present in most of the cultivated tomato varieties in Europe. And we are currently characterizing and introducing into elite lines resistance against the Rugus virus, which is compromising tomato production all over the world. The improvement of flavor and health composition is also well underway, with advanced materials aimed to contain higher levels of the good volatiles contributing to good flavor. In Europe, we normally take tomatoes for granted, but they are under constant threat, and we need to make sure that we develop varieties that are resilient and able to produce and satisfy all different stakeholder needs. So all stakeholders, from consumers to producers, will eventually benefit from our innovation activities. Breeding companies and farmers will be the most direct users of our pre-breeding innovation. And also scientists, because additional knowledge and the database are powerful instruments for future work.
Hi, I'm Giovanni Giuliano, leader of Work Package 4 of the Harnesstom project. The general objective of Work Package 4 is to develop molecular and bioinformatic tools and protocols to increase the speed, efficiency, and precision of breeding. This work package has 5 main objectives. The first objective is to increase the generations that breeders can grow in a year. This objective has been largely achieved through the application of particular physiological conditions like fertilization, temperature, and light regimes, as well as of in vitro embryo rescue methods. The second, and probably most ambitious objective, is to find genetic methods to increase recombination, to increase genetic recombination. Given that breeding of new varieties is based on crossing, followed by reassortment of favorable genes, this objective, and the first one, will decrease the time needed to generate new varieties that are more productive, tasty, and/or resistant to pests. Some genes derived from wild tomatoes that increase recombination in domesticated tomato have been discovered. We are now testing their applicability to industrial breeding. The third objective is to sequence or genotype with cost-effective methods the genetic materials generated by project partners thus facilitating marker-assisted breeding. Several hundreds of materials have been characterized by different molecular methods, generating hundreds of novel associations between traits and molecular markers that will facilitate breeding for those traits. The fourth objective is to generate computational methods able to select for a given trait, for instance, yield or shelf life, at the whole genome level rather than at a single gene level, using molecular markers. These so-called genomic selection methods have been tested with test populations developed in the Harnesstom project and have been shown to have a good prediction accuracy, all using only few hundreds of molecular markers. And finally, the fifth objective is to collect all the different genetic materials and populations developed in the Harnesstom project into an accessible resource and provided the data generated to the Work Package 3 for inclusion in the project public database.
Hello, my name is Yulin Bai from Wageningen University in the Netherlands. In this EU project, I lead the Work Package 5. The main objective of this work package is to improve resistance of tomato to 3 diseases. The first one is tomato leaf mold diseases. Second, tomato brown rugose fruit virus. And the third one is tomato leaf curl nudilli virus. We make use of tomato relatives to identify genetic resources of resistance. And so far, we have screened hundreds of tomato wild relatives and identified novel resistance sources. Tomato genes controlling the resistance are being identified and cloned. Further, we are introducing the identified resistance genes from wild tomato into cultivated ones. At the end, we will deliver pre-breeding materials that have resistance to these 3 diseases, also molecular markers that are associated with the resistance. These materials and markers will be used by breeding companies to breed tomato cultivars with resistance, and farmers will benefit from these new resistant cultivars. Finally, the consumers will benefit from the tasty tomato that are harvested from greenhouses or fields where chemical control are not needed to control the diseases. Our results will also contribute to the scientific understanding of the molecular interaction between plant and pathogens. We are in good progress to make the project successful and to deliver what we have promised in the proposal.
Hello, this is Antonio Monforte, and I'm going to update you about the progress of the work package 6 of Harnesstom. The main objective of this work package is to develop new tomato varieties adapted to drought, temperature, and selected stresses to contribute to sustainable agriculture in the near future. To address this challenge, we have formed a collaboration between academia, non-profit associations, and industry partners. The academic partners have firmly identified yeast or QTLs or rootstocks that increase tolerance to various stresses, then will be transferred to elite cultivars using marker-assisted and genomic selection breeding programs. Additionally, we are conducting fine mapping to identify stress-resistant genes to facilitate the marker-assisted programs. Currently, we are fine mapping QTLs involving tolerance to abiotic stresses as incidence to blossom end rot, fruit setting, stigma exception, and pollen viability at high temperature, and yield and then drought stress. We have identified candidate genes for some of them. Furthermore, we have verified effects of heat tolerance QTLs that are stable in an array of diverse elite tomato cultivars. So these QTLs can be used to improve the heat tolerance of a wide range of tomato cultivars. From now to the end of the project, our main objectives are to robustly define candidate genes associated with tolerance QTLs and to evaluate the performance of new breeding lines carrying these QTLs in diverse paleoclimatic regions. By achieving these goals, we aim to further enhance our understanding of abiotic stress tolerance in tomatoes and provide valuable tools for the development of resilient and high-performing tomato cultivars.
The main objective of Work Package 7 is to develop tomato lines that have improved fruit quality traits, mainly flavor and health-related compound composition, while maintaining characteristics of interest for producers and other stakeholders, such as productivity, shelf life, and good performance under different conditions. 4 tasks were performed or are underway. In the first task, CSIC and INRAE analyzed a total of 32 studies from the research performed in different countries. For each trait related to fruit quality, the most favorable QTL regions, alleles, markers, and genetic resources carrying them were identified. Data were organized and provided to Work Package 3 to be entered in the Harnesstom database. UNINA selected 14 studies analyzing tomato volatiles together with flavor and aroma, and they identified about 100 volatiles which are important contributors to tomato aroma. The second task consists in improving sweetness, acidity, and volatile composition by the introgression of QTLs into elite materials. Based on the information from the previous task, pre-breeding lines are developed. In CSIC, they started, for instance, by validating and fine mapping 15 QTLs carried in 6 introgressed regions, and they tested their impact through sensory analysis. This year, they focused on the 2 most interesting regions and started characterizing the QTLs. The other partners, INRAE, Gauthier Semences, and Villemorin, started marker-assisted selection after selecting different elite materials and donor lines and breeding regions. They performed several backcrosses and used marker-assisted selection on hundreds of plants. In parallel, UNINA used new protocols to assess the volatile profiles of breeding accessions of semi-ortho and correlate the volatiles with sensory data. These materials will be tested by sensory analysis and volatile content next year. In the third task, ENEA and INRAE are presently studying 2 F2 populations for shelf life and health-related antioxidants, and QTLs will be mapped from this experiment. In the fourth task, we are optimizing pre-breeding methods for several quality traits simultaneously. A population for the long-term improvement of several quality traits is currently under construction by Villemorin and INRAE. It will be studied next year for fruit quality traits and genome analysis. INRAE is also analyzing the most appropriate genomic prediction models in order to predict the best performing accessions. Finally, the original materials and the introgressed ones will be assessed in different growth conditions next year in collaboration with CTAX. Thus, this Work Package 7 will provide new information, but also molecular markers, protocols, and plant materials to breeders for the future creation of tomato varieties with improved fruit quality. This should also, at the end, satisfy tomato consumers.
The vast reservoir of genetic diversity in tomato germplasm banks is an invaluable resource. This rich diversity holds great potential for addressing climate change and enabling the development of locally adapted, high-quality products. Recent EU-funded projects have dedicated significant efforts to characterizing these genetic resources. Building upon these initiatives, Work Package 8, or Harnesstom, is actively employing various strategies to overcome the obstacles in accessing these germplasm collections. participatory plant breeding, and citizen science strategies following a from farm to fork approach and involving more than 5,000 participants have been conducted since the beginning of the project. The main objective is to empower farmers, chefs and citizens to explore and use the diverse genetic materials available within these collections. The collaborative efforts of the 10 research centers involved in Work Package 8 are beginning to yield interesting outcomes. A survey conducted with over 3,000 Europeans has allowed us to classify consumers into 5 distinct sensor clusters. Simultaneously, we have compiled and reanalyzed passport, phenotypic and genotypic data from more than 3,000 accessions. Based on that, we have created a repository comprising 241 superior genotypes that exhibit significant potential for direct utilization by farmers. The repository has been shared with farmers through the distribution of plantlets and with citizens through the organization of citizen science experiments. In the coming months, we will actively promote initiatives facilitating the transfer of materials to farmers and consumers. Specifically, in the summer of 2023, we will invite chefs to participate in the sensory evaluation of the collection, and will organize a tomato phenotyping training course open to a general public. Looking ahead to 2024, we have planned to unveil a range of improved land races that display genetic resistance to different pathogens. These activities reflect our commitment to fostering collaboration between researchers, farmers, consumers and chefs to promote the cultivation of diverse tomato varieties. All this information will allow to design a pipeline for efficiently transferring the results of research projects and to reinforce the open access condition of the germplasm tomato repositories.
Work Package 9 is focused on the assessment of achievement of Harnesstom project. Our main aims is to evaluate the impact of our outcomes on the market, on the breeding company, on producer, and we started collect the information about key outcome, including classification, application, and infrastructure needed to implement them. And then we conduct several surveys to understand if there were innovation of interest for the stakeholder participating to the project. Most of the innovation were of interest for the breeding company, and also the pre-breeding material was quite interesting, and at the moment is ongoing in evaluation for the seed market of this pre-breeding material. At the same time, we are performing a life cycle assessment of the breeding process by itself. I told you the material that is interesting, as well the process that could be of interest for breeding company, were evaluated to understand if they can use, and at the moment the breeding company are aware about the training that the personnel needed to use them, as well infrastructure that they have to implement. And the pre-breeding material that is under development is very promising, and we are sure that in the near future will be ready for the market.